little bit of shop talk for today. So as I said, if you ordered your book through Connect, open it, make sure it is the correct book. We've had one student already get theirs and it is the wrong book. Somebody packaged it wrong or what, and I'm hoping it's an isolated incident. So the other thing is, as far as Connect is concerned, I hadn't updated all the dates yet. I just quickly shifted them when I copied from last semester to this semester. So I've updated those yesterday for you guys. So some of you guys may have looked at it and said, oh my God, are all my assignments due like next week? No. Right? Yeah, so that's fixed. I fixed that yesterday when another student came by and said, oh my God, there's no way I'm going to get all that done. I said, no, I'm behind. Let me fix it right now. So I did that. And Canvas is still relatively new for me and for a lot of us too. So I haven't posted all your exam dates because I couldn't figure out how to change something, and so of course I did like they tell us to do. I clicked the help button in Canvas and I typed in what I was having trouble with and it told me how to fix it. So I fixed it for my lab class. You guys are next. So pretty soon I'll have all our tests listed with the due dates, which will also show up on your calendar, all that cool stuff. The other thing you may notice is that you may already see some of that when you go to your grades, because again I just copied my course from last semester over. So all those dates, I didn't know how to change it massively. I thought I clicked the button to do it. It just didn't do it. Could be user error. I don't know. So I'm in the process of, updating that for you guys. So don't panic about any of those dates. But at this point, you should have finished the introduction discussion board, right? If you haven't, get on it, because I'm going to be grading like crazy the next couple of days. I have over 100 students, though. So I tried to get ahead. So some of you guys, I kind of graded you as you guys posted at one point before I got crazy busy with other stuff. But you're past the deadline now. So if you don't get it in before I grade it, the chances of me grading it, not so good, right? Because I'm not going to start over. If I graded before the deadline yours and you hadn't finished it, I, of course, am going to regrade it, right? So if you had done your initial post and you hadn't replied yet, and you have replied now, then I will go back and give you your credit. Does that make sense to you guys? Okay. So make sure if you haven't done that, do it. The other thing is obviously you need Connect to be successful in this class, right? And then you get the special deal of the cheap book. And most of you guys, when I looked Sunday night, have at least done courtesy access. A whole bunch of you guys have actually licensed or paid for it because you want to get your book, which was great. It's one good thing about this. The bookstore will probably hate me, but that's okay. I hate them too. I'm at war with them. They mark up the prices on your books, and they didn't order my lab manual this semester. So. My students don't have their books yet. Anyone been to the bookstore yet today that's in my lab class? Okay. I'm actually about to go. Yeah, I'm going to go after. Yeah. I don't know. I checked yesterday. You didn't get a message from me because they weren't in. And she was like, 2 days shipping does not include the weekend. And I was like, oh, most of the world did at least include Saturday, but apparently not.
Yeah, I ordered mine through the bookstore, and they're supposed to send me an email, and I don't think I've gotten it.
You haven't gotten it yet?
Yeah, I didn't get anything.
Okay, good. So this is a new thing we're trying with them, too, because the other issue, y'all, if you're not in my lab class or another one, go online through their bookstore and reserve your copy, because they only ordered 50 copies, and we have 68 lab students. So somebody's going to be waiting even longer for their book.
Is that the link that you have on there?
Yeah. Yeah, and actually I turned it on in Canvas for my lab students. I don't know if other instructors did, but there should be a link in Canvas that says order textbook. And then you go to it, you put in City Park Campus, Science and Math, Biology 212, all sections. It'll come up. Right? So it is what it is. I'm not happy about it. Doing the best I can. Okay, so, but you really got to go and reserve your copy, right? And obviously we'll help you out in the meantime. So as far as Connect goes, right, we haven't really finished Chapter 1 yet in this course. When we do, study up, do those questions. Now, I had a student of mine that I saw on Saturday. I'm not in a reiteration, so these assignments I've been doing for a while. There shouldn't be any mistakes, but that doesn't mean there isn't. You guys no longer can report if there's a problem in Connect. You have to let me know and I report it. So if you think something's not grading right or something like that, please, if you know how, take a screenshot or take a picture with your phone and send that along with your email as to what's going wrong in Connect. And I'll do what I can to get it fixed. I don't think there should be any problems with this set of assignments because I've been using them for several semesters now. I hand-pick my questions. These go with our objectives. These are an excellent study guide for you guys as you're preparing for your tests. Once we finish a topic, you study it. Take those assignments as if they're quizzes. Close your notebook. Close your book. See what you actually know, right? Hit submit. Once you submit, it's going to tell you the answers, and you can take it again, right? So study up. Take it again. If you're going on the third time around and you haven't figured this stuff out, you might want to come see me, right? Because obviously you're not using a study method that's effective. You guys are all at least second semester students. You should hopefully know what works best for you. You know, students are like, how should I study? I said, if you've gotten to this point, you should hopefully know what works. If you don't, come see me, right? And I'll give you some other things you can try or send you in another direction or get you different help, right? We all learn differently, right? There's not one cookie cutter way I can tell you to do it. Other than study often in little chunks. That has been pretty proven for everybody, right? That's pretty much a mainstay. Either now or before you leave, make sure you write your name on the roll. So, as I said, handpick those questions and those assignments so they really do match with our course and really will help you know if you're ready to take a test. Okay? The other thing that people have been asking me about is an agreement assignment they may have seen listed. That's going to be next week. I try to layer technology on you guys 1 week at a time. We'll be using a special program for testing, and I want to make sure you guys have access to it or download it to your own computers. And so in way of doing that, I link that up with my agreement assignment, which is basically just a syllabus quiz, right? So you know your responsibilities in this course. But you also know how to access that software that you'll utilize for testing and also you'll utilize for reviewing your testing. So we'll go to the Hibernia Center, which is in Building 1. And I like that computer lab a lot because it's close to my office and my lab and stuff. And they're really great and nice in there, too. They have the software there on their computers called Lockdown Browser. So do they upstairs in the Big Fishbowl, the open lab on the second floor of this building, Building 2. They have the Lockdown Browser software icon on theirs as well. You have to use that to be able to view your tests. One, you'll have to use it in the lab to take the test, but afterwards to see your results, you will have to use that program. So you either could download it for free at home when I give you guys the link, or you have to go to one of those 2 computer labs that have it installed on the computers to be able to review your test results. You cannot print. There's a reason for that, right? So you can't quickly, easily share my tests with other people, right? Then I have to make harder, more difficult tests. But this is for you, so if you're having to write down what you missed and what you need to restudy for the final, it'll help you learn it too. So I'm actually helping you by allowing you not to print, right? Make sense?
Yeah, I already downloaded Respondus on my laptop for a different class.
Mm-hmm. It'll still work. Because when you download Respondus, if you're using it for another class, you log into Canvas, right? It takes you right to Canvas, you log in, then you just choose which course you're going to. So it's the same program. So you're one step ahead of everybody else. Yeah. Okay. Any other shop talk stuff? So I talked about Connect and that. Talked a little bit about some stuff coming up. Reminded you guys about discussion boards. And I physically have to grade those and we'll be working on those soon. Anything else I forgot? Remember to check your book when it comes in. Make sure it's the right book. Okay. So let's roll. All right, so let's go back to microbiology. So we just briefly, I think, started to introduce Anthony von Leeuwenhoek and talking about how, sometimes he's credited with being the father of microbiology because if it wasn't for the ability to see these microorganisms, which he was able to do by making his own lenses, his own little rudimentary microscope. And the reason why we know he did this? How do we know he did this? Well, he's credited with the first to actually see microorganisms. Whether he was the first to actually make a microscope might also be the truth, right? He kind of made his own little microscope. But how is it we know he did this?
Did he do it with like a cork an old cork from a wine bottle?
Right. He actually, the first few things that he looked at, he was a drapery merchant. So he actually started looking at his fabric under a lens that he made to be able to see how good quality it was, right? The fabrics that he was buying. But he was an inquisitive individual, so he didn't stop there. He looked at things like cork, and he looked at his own poop even. He looked at water, pond water, right? So he looked at all these things, but how do we know that he looked at all these things? Well, come to find out what I've been reading recently, he didn't actually do this drawing. He hired an artist to do it. But he drew it, he wrote about it, and he sent letters to the Royal Society in London. We still have these documents. He documented what he saw, right? You don't get credited with anything unless you have evidence, right? And the most common form of evidence in any type of science is documentation, whether it be a scientific journal that you post in, right? Or nowadays on the internet, if we can validate it as truth. But documentation is very important. And as I said, he actually apparently hired someone to draw this figure. But he saw an organism shaped kind of like what we call a rod or a sausage hot dog shape. And he saw another one that was a little bit smaller, and he even denoted that it did what? It moved. It moved, right? It followed this dotted line path. This isn't actually a semicolon. He actually saw two little round organisms. He saw a much longer rod that was had a little bit of a curvature to it. And various other shapes and lengths, even a bent rod. So, we obviously couldn't start this debate until we had the tools and the ability to see these things, right, that Anthony allowed us to go there. And there was this belief, before we had the ability to start really having the tools and the ability to ask these questions, in a phenomenon known as spontaneous, I'm thinking of genetics, spontaneous generation, right? So what does that mean? What is? Yeah, that stuff could just poof out of nowhere, like magic, right? And more specifically, that living organisms, like bacteria, could develop from non-living decomposing matter, right? So that a hamburger steak can turn into bacteria. We now know that's not the case, right? Your orange juice or your apple juice is translucent when you buy it from the store, right? Apple juice, you can see through it. It's not cloudy. Okay? If it gets pushed to the back of your refrigerator and you forget about it for a while and then you pull it out and it's cloudy, you going to drink it? No, because what happened? Something's growing in it, right? Probably microorganisms to such a large number you can see them, right? It's a huge mass, it's cloudy. Okay? How did they get in there?
When you opened it?
When you opened it. And if you're in a household where someone decides to drink right straight out of the container, right? In that case, I put your name on it, it becomes your container. Your saliva, right? Whether you backwash or not, just touching your lips to it, you've just contaminated that vessel. And that's food. Just as it is food for us, it's food for microorganisms. What are they going to do? They're going to eat it and they're going to grow. The problem is that do microorganisms make us sick?
Pathogens.
Yeah, a lot of times. So do you know if what grew in that apple juice is going to make you sick or not? No, and a lot of times when microorganisms eat our food, they convert it into things we don't like. This is when we say it's caused our food to spoil, right? On the other hand, though, sometimes we purposely put microorganisms in our food to turn it into something else that we do like. Things like what?
Dairy products.
Dairy products, right? Dairy is very commonly changed, fermented by microorganisms into other products we do like to eat. Cheese, yogurt, buttermilk, sour cream. We can turn barley and hops into things like beer, right? We can turn grapes into wine. These things we do like, right? But can we turn it into stuff we don't like? Absolutely. Can we have potential microorganisms there if you ingest it that you're going to get sick and die? Absolutely, right? And that's the fear. So they recognized, right, that these living organisms were there. But they didn't know, because they were so tiny, that they were there to begin with. It was just when they grew to large numbers we really recognized that they were there. Right? So they just thought, apple juice, poof, could turn into a whole bunch of bacteria. Not the case. Bacteria were introduced into that apple juice and grew to large numbers with that food source. So I'm going to back up for a second because I skipped over these guys. So there are actually 6 different guys, but 5 particular lists here. And the reason why John Tyndall and Frederick Cohen are listed together is you notice they're around the same time frame. They discovered the same thing, right? So they're kind of both lumped together. I have dates on here, not because this is a history class or for no reason at all do I expect you to memorize these, right? Apparently the door does lock. Try to knock. It's one of those days today. Okay. Just to give you an idea of how long ago this was, right? I don't know about you, but when you say 1626, I have trouble even fathoming. I can't even imagine that. Can't even. I mean, this was, feels like forever ago for most of us, right? So this is a really long time ago. That's the perspective I want you to see when you see these numbers, right? And really how long it took, too. I mean, look at, looking 1600s, it wasn't until, the early 1800s. That's 200 years, y'all. 200 years they were debating this, where this stuff was coming from. So we have several scientists. You do need to be able to recognize their names. You don't need to be able to spell them. Or say them. That's a whole nother nightmare, right? Some of them are not easy. So we'll start out with the first one, and his key experiment. Francesco Redi. And so he did a not very complicated experiment, right? But very thorough. He took several containers, and he did this in duplicate, right? So that, that way you didn't have, this extraneous thing that happened in one vessel and didn't happen in the other. So he took meat, he exposed it to the environment, to flies. And the flies, of course, do what? They lay eggs. Can you see the eggs? Not unless you've got really good eyesight. Right? Most people, right, don't recognize that the eggs are there, right? You may need something like a magnifying glass, right, or really good eyesight, really good observation skills to notice that the eggs are there. But over time, those eggs hatch into what? Maggots, right? These larval stages of the fly. These are quite large and visible. Why does the mama lay her eggs on the meat? It's a food source for her babies when they hatch, right? So, of course, if they didn't notice, right, that the flies laid the eggs, they thought that these maggots just spontaneously generated out of the meat. The meat turned into maggots. We now know, of course, that's not true. But to help prove that, right, he allowed that to happen, and then he covered up the meat, right, and found that it just continued to decay, no maggots formed. Why?
Because it was covered.
It was covered. The flies could not get to the meat to lay the eggs. But then some scientists will argue too, well, there were certain things that weren't allowed to get in because you sealed it up that stopped the growth. So what is something that some organisms sometimes need for growth? Air, right? So in this one, he covered it with gauze, right? So air still got in, but the gauze had tiny enough holes that the flies could not get through. But instead, the flies could still smell, right, that food source nearby. So they laid their eggs on top of the gauze, and then you got the production of the maggots. The bad news for the poor maggots is they couldn't get to the food, right? So with this experiment, he could really show, right, that those maggots were coming from the flies, right, and not spontaneously generating from the meat. But those are relatively large organisms. And like I said, some people, right, really good observation skills, really good eyesight, might actually see those eggs. So that was for relatively large stuff. But could this be true for microorganisms as well? So imagine that apple juice container, right? How are we going to prove something you cannot see until it grows to those large numbers is present in the air or in our saliva? And that's how it's getting in there. So we've got to, try and prove that, right? Scientists that believe that it was not just coming out of the food, right? The food was not turning into these organisms. It was coming from somewhere. So John Needham started out with a food source, right? Some type of liquid food source, maybe a yeast broth, or imagine something like chicken broth or apple juice. If you boil it, what could potentially happen to any microorganisms that are present in it?
Destroy.
You could kill them. Is this true for all microorganisms, though?
No.
Now, we know nowadays that boiling is not sufficient to render something what we call sterile, completely devoid of living organisms and viruses. But most of the time, it will kill most microorganisms. And in fact, we do a really quick heat, and there's different types of protocols for this. That apple juice that I talked about before you put it in the fridge, where was it? It was on the shelf, right? You probably bought it right off the shelf in the grocery store. Put it on your shelf in your house until you wanted to drink it, and then you opened it and you put it in the refrigerator. So why is it that you were able to keep it on that shelf? It was pasteurized, right? Most juices are pasteurized to eliminate microorganisms. One, for the potential of disease. Two, to increase shelf life. The less microorganisms that are present in it to begin with, the longer it can stay fresh, not spoiled, or not make you sick. If you pasteurize something, does it mean it has no microorganisms at all?
No.
No, it's significantly reduced, and it depends on which pasteurization process. Right? So things like milk can be pasteurized to a level that you can keep it on the shelves. Right? Most Americans do not like this type of milk. Right? It changes the texture, the content of the milk. Right? Different from what we're used to. Where in Europe it's very common, right, to have ultra-pasteurized milk that's not refrigerated, right? So some Europeans come here and they're trying to find the milk that they're used to and they can't find it. And then, of course, we go over to Europe and we can't find the milk we're used to, right? And it just has to do with, how we preserve things, right? We can do it in different ways. So imagine a food source, right? Maybe that apple juice, right? We've boiled it, we pasteurized it, we've removed most of the microorganisms. But if you leave it open to the air, what's going to happen? Microorganisms are going to get back in, right? They're in the air we're breathing right now. They're going to fall in. That's food. So what are they going to do? They're going to grow, right? When microorganisms grow to large numbers, can you see them? Yeah, it becomes a cloudy liquid. There's growth in there. So did he disprove spontaneous generation? No, he kind of helped support it, right? He's like, look, even after I boiled it, it can still turn into microorganisms. No. First experiment's John's. Next experiment is Lazzaro. I can't even say his name today. Good old
Lazzaro?
Lazzaro, thank you. So Lazzaro went one step further, right? He heated, but then he sealed the flasks, right? So hopefully he killed the microorganisms by heating them. He sealed it up so no more got in, which is what we typically do with our juices, right? When they pasteurize it, they seal it. We don't have to worry until we what? Open it, right? So no growth until he opened it. So obviously, right, now we're going, okay, something is getting in. But still others were like, okay, well the problem was, why do we get no growth here? It's kind of the same problem that Redi ran into. No air, right? But we know today, can microorganisms grow without air? Some of them can, right? You can suck the air out, right? That's the way we preserve some of our foods, is to suck the air out. That works great for aerobes, organisms that require oxygen. The ones that don't require oxygen, you just made them really happy. Right? Those good old anaerobes. So there isn't one fix for all things, right? There's no perfect way to preserve. Right? There's different methods used for different reasons under different conditions. So we still have this problem though, right? We still have this problem of air and growth, and how do we keep the air clean? So how do we make sure that the air is clean but still gets in? So how do we do that? Pasteur helped figure this out.
Special glass tube?
Before that even. So think about in your house. What helps clean the air? What do your air conditioners have? Filters. Filters. Even your cars get an air filter, right? We want to filter the stuff out. So he used cotton, right? And filtered the air. And he took that cotton that now had the trapped microorganism that you couldn't see and put it in a broth, and again, he got growth. He's like, there's something in the air. I trapped it. I put it in here. Look, it grows. But again, that wasn't enough. Never is, right? So he's like, okay, I'm going to show you in a way you can understand. I'm going to do sterile broth in a container, but this container, I'm going to stretch out the neck. I'm going to bend it. And by doing so, air could still enter, but indirectly, right? We use this technique a lot in microbiology lab, right? The first week, I told all you guys when you were dealing with the plates, where should the lid be? Right over the plate, right? So that you're not breathing on the plate that you're inoculating, so that stuff is not falling out of the air onto your plate, right? So that you can sneak in from the side, right? And that lid is helping to protect. When we deal with our tubes, we hold them at an angle so that airborne contaminants aren't easily dropping in. So again, the air is coming in directly, so what happens with those contaminants? I mean, is the air is going to move, right, through, but those contaminants are going to stick to the sides as it's going through. Can you visualize that? See that? So even though those are open to the air, because it's indirect, unless you were blowing it in and forcing that dust up and over, it's going to get trapped in those beds. So he was able to literally for years leave flasks on his bench top, on his shelves, and no growth occurred. And they're like, well, of course, the other one's like, well, maybe that food just cannot grow. That that's inert food. It's not good food. Nothing wants to eat it, right? So he's like, okay, I'm going to show you. And he broke his flasks and exposed them. Or even tipped them and exposed them to the stuff that got stuck in the bends, and guess what happened? Growth occurred, right? Because those microorganisms that we can't see with the naked eye are present in the air. Right? So anything exposed to air with dust in it, with contaminants in it, could potentially grow microorganisms. So they have places, right, where they have special filters, clean rooms, right? No dust at all. Nothing will grow in there. You could leave the flask completely open, right? No indirect. But there's no microorganisms in the air. It's clean air.
And it was due to the filter.
Yeah, filtering it out. HEPA filters stands for high efficiency particle air filters. They're very, very tiny holes in those filters. Most of the stuff gets trapped on the filter itself, and only the air gets through. You can buy them for your home. They're great, right? Especially people who have allergies to things in the air and stuff like that. So they fill up fast too, especially if you have a kid that goes in and out the door all the time and dogs that go in and out the door all the time. It's really not cost effective in my house. My mom, it's just her. She buys the expensive HEPA filter. It'll last her a month. Me, it'll probably last like 2 freaking days before that thing is full and no air is getting through. Right? It's filled up with stuff. The biological hoods that are in the science labs, right, where it's creating a clean air environment. It's HEPA filtration, right? Really good filters filtering that air that goes through that laminar flow hood to keep that a sterile air environment. So you don't get contamination of the cultures that you're working with within that cabinet. But it's not closed, right? There's instead a curtain of air circulating through it. So we have one now in the biotech. So if you're in microbiology lab, if they're open in there, we'll sneak across the hall and you can see a flow hood. In the microbiology lab, we don't need that clean of environment. But again, we're going to work in a particular way that we're going to try and avoid contamination of our cultures, right? We're not going to breathe on them, right? We're going to try and limit their exposure to air because we know there's microorganisms in the air. So was everybody happy with this? They're like, okay, Louis, we believe you. Let's move on. No. Some people tried to repeat his experiment. And you'll see if you read scientific papers, nowadays they'll tell you exactly what brand of Petri dish they used. Maybe even the lot number. Right? Exactly what they used in their experiment so that if you go, because you don't believe them, right? So you're going to repeat it and be like, I got something different, right? Guess what happened? Some people tried to repeat his experiment. Did they do exactly what he did? No. Some of them use a different food source, a food source that is commonly contaminated with microorganisms that are heat resistant. Boiling does not destroy them. We talked about a disease at the beginning of this course, which was last week, right? That's caused by these very heat-resistant forms known as endospores. What disease? Anthrax. Right? So if those spores get in and you boil, do you destroy them? No. So you're still going to get growth, right? So some of these guys might have gotten some endospores in there. Boiling didn't destroy them. They still got growth. So like, Louis, whatever. So it wasn't until we knew about the endospores, and that's thanks to two gentlemen working around the same time in two different areas. Again, they continued on Louis' research. They demonstrated that dust in the air is actually what's carrying the microorganisms.
For the Lazzaro's experiment,
Mm-hmm.
why wasn't there any anaerobic?
Yeah, and again, conditions are going to really depend, and that's why in scientific experiments nowadays they're very specific under what conditions. So he asked, well, why didn't any anaerobes grow when he had that closed jar, Lazzaro? When he had that closed jar, well, because none were in there, right? And probably his boiling, if they were, he did kill them. But there are some anaerobes that produce spores, like what causes botulism, right? The organism that causes botulism is a spore former. It can get into your cans. Once the air is sucked out of those cans, then it germinates. And again, it's a spore former. So would a boiling killed it? No, he just got lucky. He didn't have any endospores in there. And same thing with Louis. His experiments would have been completely different had his food source he chosen been contaminated with endospores. A totally different story. So it wasn't until these two gentlemen, right, continued on his research and actually found and proved that there are extremely heat-resistant forms of bacteria that you cannot destroy with boiling. But, as well, it really is the dust in the air. So if you could filter out the dust, right, you could keep air clean, you're not going to get growth. You're not going to get contamination. So, Ferdinand Cohen and John Tyndall. And John Tyndall, there's a process for eliminating endospores without using modern techniques like an autoclave, which is high-pressure steam, which can actually destroy endospores. Other than that, you could boil, and again, you're going to kill the ones that are in the vegetative state. And then you could incubate with food, and those endospores will germinate, right? Because they have food, they're not under harsh conditions, they're going to germinate into vegetative cells. Then if you boil, you're going to kill them, right? Because they're vegetative cells, they are susceptible to boiling. And then just to be sure you got them all, you would do it one more time. You would incubate again. Hopefully all your spores have germinated at this point. So when you boil again that third time, you should hopefully have eliminated all the organisms in that sample, including the heat-resistant ones. That's called titillization. And of course, that takes about 3 days. So I don't know about you, but, 15 minutes or half an hour in the autoclave, I'll trade you. High-pressure steam, baby. Take care of that in no time. But, if you don't have access to that type of technology, there is a way to, over time, eliminate those endospores by encouraging them to form vegetative cells which are sensitive to heat, like boiling. Make sense? So you guys just didn't even realize how much you already know, huh? About microbial growth and why it happens and how it happens and why we don't want to eat certain things and why they smell bad and why we do the things we do. But it was thanks to these guys, right, that we have that level of understanding. And then as it relates to microorganisms, of course, some are really good, right? They make the beer, the wine, the cheese. Others are really bad news. They actually cause disease. But even when it came to that, when somebody got sick, and I'm not kidding you, people believed, and some people even to this day, believe that a deity or God or something is punishing you. And that's why you have this disease or why you're sick, right? A lot of us nowadays know, right, something is potentially making you sick. A bacteria, a virus, a fungus, a genetic defect you may have been born with, right, is why you have a problem or a disease. But we had to be able to prove this too, right? They had to be able to prove if you've got anthrax, it's because you ingested the bacteria or the endospore of that bacteria. That's how it is you ended up with that disease, or you inhaled it, right? Or it infected a wound. you got exposed to this. But again, they're microscopic. Do we see the bacteria in this air we're breathing? Do we see the viruses? No. So we have to have some way to see these things, test these things, prove these things. And so it wasn't really until we were able to do that this level of understanding and our areas of study expanded, right? This understanding of the immune system, right? Why is it that some people get sick and others don't? So again, it wasn't obvious right away. We had to be able to test and know these things, right? To be able to establish those connections, we need to develop the techniques that we have today to be able to study. So those Petri dishes that I'll bring into lecture for those of you guys not in lab next week, so you can actually see, right, that specially designed, they used to be glass, now they're plastic, container that we use to hold the food and grow microorganisms. Even the food itself, how it's prepared, how we create that solid surface within a Petri dish is due to someone discovering that a polysaccharide found in marine algae called agar can be used as a solidifying agent. Otherwise, we'd be growing everything in broths. But, you know, organisms grow differently on surfaces. Imagine the difference of growing an organism on the surface of your orange as opposed to in your orange juice, right? That's a totally, completely different environment. One is aqueous. If it's a motile organism, it could swim, right? One is a solid surface. And so how the organisms grow, how we're able to study them, really is dependent on having an ability to grow them, for one, right? And grow them on surfaces as well as in liquids, okay, if necessary. And we need a surface that is going to stay a surface, right? As they eat the food, that surface doesn't go away. And that's the good news with agar, is that we haven't really found a microorganism yet, although I'm sure there's probably one out there, because there are ones that eat oil, so there's got to be somebody who eats agar, right? But so far, none of them will eat the agar. They'll just eat the nutrients. So the platform, that solidifying surface stays, even though they eat the food that we put with the agar. So most of us are familiar with gelatin, right? That's a protein that under certain conditions will become gelatinous or almost solid. But at room temperature, as we know, what happens to it? It melts. So one problem with using gelatin is that we can't incubate it at high temperatures like body temperature. It would melt. So again, not a solid surface. The other problem is the protein. And just like us, a lot of bacteria like to eat it, right? So they would make it into goo, and it would no longer be a solid platform. So agar was a really great advent to being able to grow microorganisms on a solid surface, right? Without that surface deteriorating under warm conditions or when things were being eaten. The Petri dish itself is designed in such a way that air can get in indirectly, right? So that we don't get airborne contamination of our cultures, but microorganisms that need air can get air. Same is true for the tubes that we use, unless it's a screw cap, right, where we want an anaerobic environment, right? Other ones allow air to come in, but again, indirectly. So, as I said, once we figured out that microorganisms cause disease, we could go one step further even and talk about why some people get sick and others don't as it relates to our immune systems. So, Robert Koch, working underneath his teachers, developed a set of protocol to determine this relationship, right? That Bacillus anthracis, that bacteria, causes disease we know as anthrax. And as you can tell, the name of the disease, came before the name of the organism. So once we discovered the exact organism, it was actually named after the disease, where that happens a lot, right, as far as naming goes. So this set of criteria was developed by his teacher, right, and he followed through, right? He did the legwork. So it is now known today as Koch's Postulates. It's still used today sometimes to determine the link between a particular microorganism and a particular disease. It can't always be used, and we'll talk about why. What are some of the constraints of using this particular protocol? So how do these postulates, how do they help us link, right, the organism to the disease? So again, I steal pictures from other books, so this one might not be the one in your book, or it may be, I don't know. But you probably have one similar. This is the one I like, though. So imagine you have a sick animal, this poor little mouse. He has some type of infectious disease. Something infected him, maybe a bacteria in this case. So they take a sample, they look at it under the microscope, they confirm we have some type of microorganism, potentially a bacteria present here. So the next step they're going to do is they're going to try and isolate it in culture. And this is where, again, solid culture media like this Petri dish come in handy. Because if you have a mixed culture, you have multiple microorganisms from that original sample, we could spread them out on this surface and hopefully isolate them from each other. So we can isolate out individual microorganisms. Now this is dependent on the fact that we can actually get them to grow on these Petri dishes. Can all microorganisms be grown in artificial culture? No, right? Mycobacteria leprae that causes leprosy? Can't do it. It won't grow in artificial culture. Mycobacteria tuberculosis will, the causative agent of tuberculosis. So they could take it out, they could isolate it, they could grow it. But how do you know that that bacteria that you took out of that original mouse causes that disease you saw in the original mouse? You've got to what? You got to test it. You got to stick it into a healthy mouse. And you got to get the same result. You got to get that same illness that the original mouse had. Now, are all diseases, can we test them on little mices? No, right? Human immunodeficiency virus is the reason why it's called human immunodeficiency virus. It's pretty specific to humans. There is one animal that's very closely related to us, and that's what?
Chimpanzee.
Yeah, chimpanzees, right? So they test things in chimpanzees as it relates to that virus, right? So we run into limitations here, right? For not everything would we be able to do this. And I had news for you. Some of the older scientists, especially in homeopathic medicine, ran out of friends because they literally would test stuff on their friends to see what happened. There has been times in our own history, right, where military members have been tested on without their knowledge. Apparently, maybe even people in the prison system. We do not openly do those types of things anymore, right? They're not considered ethical or appropriate behavior as it relates to experimentation and discovery. But some people purposely for money make themselves guinea pigs, right? Right? Some people sign up for medical trials, right? And get paid or get compensated, right? There's a whole ball of wax, and X-Files is back on. Who watched it last night? I'm the only little person in this room. It's pretty cool, though. You should check it out, young peeps. But, the possibility of experimentation without your knowledge is kind of frightening. Maybe you can't handle it. Don't watch it. Especially when it relates to aliens. So, again, we don't even stop there, right? That second mouse that we've injected, we're going to try and take it back out and culture it and make sure it matches up to the original bacteria that we isolated that we say causes that particular infection or that virus or whatever it is. So we gotta do all of this. And he did all of this for anthrax. He was able to prove that that bacteria causes the disease we know as anthrax. He went through all these steps with that bacteria. Nowadays, it's not as widely used. We can use molecular techniques, right, where we can test for the presence of of DNA from the particular bacteria and it being present and linking that to disease. Some individuals can be infected by a pathogen and yet not develop a disease, right? So what if they inject it into the mouse and it doesn't get that disease? Does that mean that that bacteria doesn't cause anthrax? Does that mean if I inject you with rabies and you don't get rabies, that the rabies virus doesn't cause rabies?
No.
No, certainly not. What potentially, hopefully, saved your ass? Your immune system, right? So that explains why sometimes, right, someone will be exposed to something and they don't actually get sick or are not outwardly sick. You don't know that they have the bacteria or virus. People can become what's referred to as chronic carriers of the pathogen. Right? My favorite story is Typhoid Mary. Right? Another one like back in the 1800s, really before we understood some of these things. She had gotten a type of salmonella, right? Bacteria. It's not a dinosaur, right? I watch too much TV now. Okay. So, this bacteria, she got the infection. She became a carrier. It hid in her gallbladder. So each time her gallbladder squeezed bile into her intestines, that bacteria went into her intestines. But it didn't cause an infection of her intestines for her. But it was excreted in her feces. And unfortunately, this woman was not taught how to properly wash her hands after going poop. And she was a cook, of all things. So a whole bunch of people got sick from eating the food she prepared. And they linked it back to her. But they didn't know how or why. The woman wasn't sick. So they quarantined her. Back then, y'all, that was prison. They imprisoned this woman. After a while, they let her out of jail. Guess what she did? The only thing she knew how to do. She went back to work as a cook. In a maternity ward, of all places. Guess what happened? People got sick. They linked it back to her. They imprisoned her again until she died. What could we have done to solve this problem and what we know about and how disease is spread? Wash your damn hands. Okay, that's one thing. We could have properly taught her how to wash her hands, right? What else could we have done?
Try to find a cure.
Yeah, finding a cure back then, right, wouldn't necessarily been maybe an option, but there is a cure for that. We could have removed her gallbladder, right? And therefore she would no longer be harboring the infection and excreting the infection. Antibiotics won't work. It won't get to the gallbladder. We could have trained her to do something else, right? Job training. I don't know. That might have been a good idea. What time do I have?
It's currently 10:30.
All right, so we're done, right? Or no, what time do we done? We have 10:40, 10:45. It's been a rough morning already, y'all. I don't have a watch. I don't have a cell phone. I have no internet. I want to go home and start this day over again. All right, so how does this observation affect Koch's postulates, right? Well, it creates a problem, right? Because, again, the mouse might not be openly sick, get the infection, right? So how might the postulates be modified to account for the existence of chronic carriers?
I guess you gotta identify the host. Like trying to see if it's latent, something.
Yeah, so, I mean, even though it's not outwardly sick, they may still have the infection. So again, we still got to try and find where it is in the body, right, and culture that. So in the case of Mary, where would we have found the bacteria? It's in her gallbladder, but what comes out of her body we could have tested. We could have tested her poop. Yeah. Right. So, and found that, she does have the organism, even though it's not making her sick. Does that make sense to you guys?
So it's like how body became used.
Well, what it is that when it gets into your gallbladder, your immune system doesn't go there. It's a site where your immune system doesn't go because your cells of your immune system would be killed by the bile. But the bacteria, on the other hand, this particular bacteria, a lot of gram-negative bacteria can survive in the presence of bile salts, and in fact, they can eat it and use it as a food source. So, yeah. So it wasn't dying, it was hiding there. You know, it was hiding from the immune system. When it got into the intestinal tract, the immune system took care of it, right? That's why she didn't get sick. But the fact that, you know, it was in that privileged site, the immune system couldn't get to it and eliminate it. And that's why, again, like we can't give you antibiotics. They're not going to get to it. It's just a storage vessel. It's not going to get inside the bile, right? You don't have blood flow to your bile, you know. So you can't get at it with antibiotics. We have to, unfortunately, remove the gallbladder itself, which means, you know, no more shrimp po' boys. You can't eat a whole lot of fat at one time when you don't have a gallbladder, because what your liver is what produces the bile. So it's always putting bile into your small intestines to help with digestion. It's when you eat a really fatty meal that your gallbladder contracts and squirts extra bile into your small intestines to help digest that additional fat that you ate. And that's why it gets stored there for, you know, when you need that little extra, right? But, you know, if you have no gallbladder, there's no more fatty meals like triple boys. So, make sense? And people who get crystals in their gallbladder, it's usually because, again, they're not utilizing it and the bile staying there for a long period of time and it crystallizes, and then they end up losing their gallbladder. Those are people who go on crash diets that eat no fat at all. And you really get a buildup of bile in the gallbladder, and that creates a problem. You do need a certain level of fat in your diet. You know, everything needs to be balanced, right?
So how would you get like gangrene in your gallbladder? Because my mom had it.
So again, gangrene is an infection. So what happened is that the bacteria got into the gallbladder and destroyed the gallbladder. And again, some bacteria, you know, bile won't kill it. So, okay, so when studying the development, as I said, it really wouldn't have been possible if it wasn't for the advent of agar, right, this solidifying agent we're using, those petri dishes to be able to grow this stuff in, a food source. And the most commonly used food source that we use is nutrient broth, which is just basically beef extract and sometimes also yeast extract. So again, it's just food, right? So, and in a form that's easily taken up by the microorganisms. And then nutrient agar, of course, is that beef extract and yeast extract sometimes with the agar, so then it solidifies when it cools and creates a solid surface. And then different methods that we use to be able to isolate out, separate out individual organisms, right? Because if you take a sample, you may get lots of different bacteria. You're looking for particular ones that are causing particular diseases. You'll use all these materials in the lab. You'll do techniques similar to this throughout the lab, throughout the semester, those of you guys in lab. Those of you guys that aren't, I post some of my videos for you guys, right? And I'll also bring some of the stuff in so you can see it firsthand instead of just pictures. So this is the last little bit. Some of these guys we've already talked about, right? What was Anthony's contribution? Anthony von Leeuwenhoek. Yeah, discovered microorganisms, right? One of the first to document microorganisms. Redi. What do you think of when I say Redi? What experiment is he linked to? The meat, right? The fact that where do maggots come from? Flies, eggs, right? And why was he doing this experiment? What was he trying to disprove? Spontaneous generation. He was part of the spontaneous generation debate. So was Pasteur, right? Pasteur, what did he contribute to this debate? What did he help us learn? Yeah, air has microorganisms in it, right? Yeah, you got to filter the air if you want it, something to be sterile. HEPA filters, right? Yeah, you found that again too, that if it's exposed to air, as long as that air isn't contaminated, right, the swan neck flask, then growth isn't going to happen. And Cohen and Tyndall really helped us with that whole problem of the ones that were heat resistant, right? So those individuals that were boiling stuff and not actually creating a sterile broth. And they took it one step further. Where are the microorganisms in the air? They're attached to the dust, y'all. They're attached to the dust in the air. What is dust? Anyone know?
Dead skin cells?
It's mostly dead skin cells, epithelial cells, and dirt. Right? Very tiny, right? Can stay within the air. So Koch we just talked about, right? Koch's postulates, what did that help us understand? The link between a disease-causing organism and that disease, right? From organism to disease. Lister, you may have something that's named after him in your bathroom. Listerine. It's named after him, right? Because he helped contribute to, yeah, he's on my last slide, to cleaning the surface of the body before doing surgery, right? He is started with this whole thing of antiseptics, right? Where we could clean with a cleaning agent. And even afterwards, what we put on the wounds had to be clean, right? So that we didn't infect the wounds after surgery. So although he didn't, he wasn't like Koch, he didn't say, bacteria caused this disease. He definitely knew there was something microscopic out there that we could be cleaner, right? That we could clean our instruments before we operated to make sure we didn't introduce these organisms into the body. That they're present on the skin, right? That we need to clean the skin before we cut into the skin. The instruments that we cut with need to be clean. The dressings afterward need to be clean. So, of course, Listerine is a mouthwash, right, that helps clean our mouths to prevent disease. I want to roll back. So, in addition to Pasteur's help with spontaneous generation, this guy's pretty impressive. He worked with several scientists. If I could get me a time machine, I'd go back and have a glass of wine with him. So he discovered that incubating cultures for long intervals between transfers cause pathogens, right, things that make us sick, to lose their ability to cause disease. This is commonly referred to as something being attenuated. We use this a lot in vaccination, right? We give you a weakened form of an organism or a virus. I call this the fat and lazy syndrome, right? Because if you keep feeding them, right, if you're growing them in artificial culture, you're feeding them, right? So they're just sitting around becoming fat and lazy, right? They don't have to fight for their food. So eventually they forget about fighting. They're like, it's good. I don't want to fight anymore, right? So those abilities that they have, they either lose them altogether, right? They don't perpetuate that ability within their population anymore because they don't need it, right? There isn't that pressure to maintain that ability to be a bad guy. I always think of WALL-E. You guys seen that movie? Right? The people up in space, they all become fat and lazy, right? Because they don't have to do anything. So then they got to get themselves back in shape to live on Earth again. Okay? The bacteria become fat and lazy. They lose their ability to be aggressive bad guys. This is beneficial to us because then they turned around and used it for vaccination purposes. Right? So Louis and others helped develop vaccines for chicken cholera, for anthrax, for rabies even. Right? Introducing a weakened or disrupted form of that pathogen into a person to get your immune system boosted, so when you got experience with the big bad thing naturally, you already knew what the bad guy looked like, and you could respond much quicker. That's what vaccination gives you, is a leg up, right? It tells you, it sends you the wanted poster. It says, this is what the bad guy looks like. Get ready for when he comes, because he's coming.
Is that like the memory cells?
Yes. That's the awesome thing about your immune system, is it remembers. When you show it something, it can remember it. That's what vaccination takes advantage of, is your ability to remember. I have 2 minutes, huh? So, vaccination against various childhood diseases have contributed to the entry of women, particularly mothers, into full-time workplace. Why? Yeah, you don't have to stay home and take care of the sick baby, right? They're not getting measles, they're not getting chicken pox, right? 1995 chicken pox vaccine came out, right? I had chicken pox when I was a kid. My generation, they brought you purposely over to somebody's house who had chicken pox. They called them chicken pox parties. So that you would get chicken pox before you went to school. So you wouldn't miss 2 weeks of school because you have the pox and are contagious. Right? You got it when you were 4 years old like me. Right? Apparently my youngest sister played very clean. Even though it's respiratorily transmitted, right? Apparently she didn't get close enough to the other kids. She didn't get it until she was in high school, missed 2 weeks of school. Right? So, vaccination has really helped us out, right, as it relates to how long and how many illnesses we deal with. It's completely changed our modern workforce as it applies to our world. Did I get everybody? Oh yeah, there's more on Louis. That's why, he's the wine man. He helped figure out the problem when their wine was getting contaminated with other microorganisms and it was turning into something not quite wine. Right? So, and again, what's named after Louis? Pasteurization. So again, that process, of helping us get rid of the bad guys and only the good guys grow. All right.